We have our guest speaker today, Lihong Wang from the University of Washington University in St. Louis. Professor Wang earned his PhD at Rice University and since then published over 250 peer-reviewed journal articles and delivered over 270 keynote plenary or invited talks. After now we will be at more than 271. He received 27 research grants as a PI with a cumulative budget of over $30 million.
Japanese yen.
He's a lab inventor or discovered over 13 different imaging and sensing techniques of phenomena. He himself is a fellow of the American Institute of Medical and Biological Engineering, the OSA, IEEE, SPIE. He's a founding chair of scientific advisory board of two companies that commercialize his inventions. He received some awards, also the First and Career Awards and the 2010 Joseph Goodman Book Writing Award for his book on biomedical optics, principles, and imaging. So, welcome.
Thank you, Ariel. Well, thank you for the very kind introduction. Thank you all for coming here. I'm really glad I made this trip. I can't tell you how excited I am about some of the research going on here. Today I'll be talking about photoacoustic tomography. Our goal is to ultrasonically break through the optical diffusion limit. WashU requires me to disclose my financial interest in every talk I give. A couple of companies commercializing photoacoustic tomography, and we're almost exclusively funded by NIH. Starting from the motivation and challenges in optical imaging, I'll be covering two incarnations of photoacoustic tomography, namely photoacoustic CT and photoacoustic microscopy. Then I'll close with a discussion and summary. Why optics? That's the very first question. You walk into a clinic, you'll see a number of imaging modalities available already. Now, first of all, it's very safe to use the light because we're dealing with non-ionizing photons. The photon energy is a couple of EVs instead of thousands of EVs in X-ray or PET imaging. From the physics perspective, which is a very fundamental point of view, that light is the only part of the EM spectrum that allows you to probe molecules directly. Right? So that means we can probe biological molecules and probe the composition of tissue directly. It starts out as a molecular imaging, even without injecting any contrast agents. Optically speaking, we image absorption or scattering contrast in most cases. Today I'll focus on absorption, which probes oxyhemoglobin, deoxyhemoglobin, melanin, lipid, even water, which is typically transparent in the visible spectral range, but if you use the right wavelength, you can look at water absorption as well. Now, recently we demonstrated we can image DNA and RNA directly. We can target DNA and RNA absorption and see cell nuclei. From a physiological perspective, we look at endogenous contrast to provide functional imaging. For example, we can look at the concentration of hemoglobin, the oxygen saturation of hemoglobin. Both are hallmarks of cancer. Cell nuclei. There's also a Doppler effect in photoacoustics or in optics that allows us to image blood flow. From a biological perspective, we can use exogenous contrast to provide additional molecular imaging of biomarkers or even reporter genes. And remember, this is a partial list. The list goes on and on. So there are many reasons why optical imaging is important. Where's the challenge? We face two fundamental challenges in high-resolution optical imaging: diffraction and diffusion. Diffraction as a wave phenomenon limits the spatial resolution of any ballistic imaging you've heard of, being it confocal, two-photon, or for OCT. People have found ways to overcome the diffraction limit for super resolution imaging. Diffusion as a scattering phenomenon limits the penetration of ballistic imaging to about 1 millimeter if you were to image scattering tissue like the skin. We want to use photoacoustics to overcome that diffusion limit to achieve super depth imaging. This movie shows you how quickly photon propagation transitions from the early ballistic regime into the later diffusive regime, right, which takes place within a matter of 1 millimeter. That's the diffusion limit defined by the transfer mean-free path. That's the mean-free path between equivalent isotropic scattering events. So we want to focus on the diffusion problem. From a very high level, what do you have to do to form a good image and still penetrate deep? If you send in light through a piece of thick tissue, the detection and the illumination and detection photon path will both be tortuous. If you want to image the middle plane, the shadow of that middle plane will be blurred. So you really won't capture a shadow like you would in X-ray projection imaging. If you were to slice open the tissue, you can expose the middle plane. Now the detection path is well defined, even though the illumination photon path is still tortuous. One-sided clarity will allow you to form a good image. You really don't have to make both sides clear. The illumination can be a turbid process, scattering process. Conversely, there are other ways to make the left side clear. But today, let's focus on the detection side. Well, obviously, that's an invasive procedure. You can't really use that for imaging. A better way is to keep the tissue intact, but optically clear the scattering by introducing osmotic agents. You can achieve the same effect, but this is a toxic process. What we want to do is convert light into sound. Acoustic scattering is 1,000 times weaker than optical scattering, so the acoustic propagation is very well defined. You're achieving the effect in B or C without invasiveness or toxicity. You're essentially listening to optical structure. The contrast is optical, but your signal carrier is acoustic. So that's the basic concept behind photoacoustic tomography. Photoacoustics as a physical phenomenon has been around for over 100 years. Alexander Graham Bell first reported photophone instead of telephone in 1880. He wanted to encode sound into a light beam, then propagate the light beam in space, convert light back into sound again. Right? So what's interesting is right here, which is the photoacoustic effect. When light is absorbed, you generate a temperature rise. Due to thermoelastic expansion, we induce acoustic emission. Of course, nowadays we're interested in the ultrasound portion of the acoustic spectrum to shorten the acoustic wavelength so we can improve the spatial resolution. Now, of course, tomography was not even in the lexicon over 100 years ago. Nowadays, we're using Bell's concept for imaging. Let me start with photoacoustic CT in a circular geometry. The basic concept is to broaden your laser beam, make sure you stay within ANSI safety limit. Typically, we use a Q-switch laser, nanosecond pulse width. We don't use unscattered photons only. Unlike all the ballistic imaging modalities where they depend on ballistic photons that have been unscattered or singly scattered, the ballistic signal will attenuate exponentially with a decay constant of roughly 100 microns. So that's why all the ballistic imaging modalities won't go deep. Here we tolerate scattering. Any photons are useful photons in this modality. As long as the photons are absorbed, they will generate some heating. Every millidegree temperature rise creates 8 millibars pressure rise, which is detectable already above the noise level. So because sound is relatively transparent in the propagation inside tissue, all we have to do is to detect the photoacoustic emission outside the tissue and form an image using the acoustic signal. However, the contrast is optics-based because when you have stronger absorption, you generate stronger photoacoustic signal. A simple analogy is triangulation of a single point sound source. Imagine there's a thunderbolt. When you see the lightning, that's your time 0. When you hear the thunder, you can record a time delay t1. Since we know the speed of sound in air, we multiply the two numbers, which defines the radius for a spherical shell on which lightning took place. If you have 3 such measurements centered at different locations, you can triangulate and figure out exactly where the lightning took place. So photoacoustic tomography is very much analogous to this, except the volumetric source now is much more complex than a point source. So your algorithm needs to be a little more sophisticated. And this is the only slide where I show you some math. There's a spherical redound transform involved. I'm sure you've heard of linear redound transformation in X-ray CT, where you integrate attenuation coefficient along the X-ray propagation direction. In our case, if you have a 3D complex object, you can bathe the object with photons. You generate a 3D volumetric sound source, and let's call that P naught, with a spatial distribution. Now that P naught is going to propagate as an acoustic wave. At time t after your laser pulse, you're integrating P naught on this spherical shell. That spherical integration is called spherical redound transformation. Although redound really never inverted that problem. A few years ago, we found a universal back projection that works for 3 standard detection geometries: planar, cylindrical, and spherical. P is what we detect on the detection surface R naught versus time. So every ultrasound transducer element receives a time trace. If you only use that pressure and properly time delay, right? Because when you receive the photoacoustic signal, there's a time delay because of sound propagation. When you back project it, you got to time delay that again toward the object. You integrate over the full aperture of the detectors. And when using the pressure only, you get essentially ultrasound beamforming. Right? That has been used for quite some time in ultrasound imaging. But if you include this pressure derivative with respect to time, you will get an exact solution. So you will recover P naught in the exact form. This is very powerful. At first glance, P naught is a mechanical contrast because that's pressure. However, P naught is proportional to the optical energy deposition. Therefore, you can reveal optical contrast through the measurement of P naught. So that's the basic concept behind photoacoustic tomography from a mathematical perspective.
Can I ask a question?
Yeah,
So
please.
You have actually a longitudinal wave because the liquid is not a sustained transfer.
Yes,
Wave.
it's a scalar wave.
So if that wave propagates through the medium, how sensitive are you to the inhomogeneities of the compressibility of the environment of that emission?
So it primarily comes in the form of heterogeneity in the speed of sound. In soft tissue, that variation is about plus minus 5%, for example, in the breast tissue. So it's not a huge variation. Depending on the geometry of the system, it may or may not be a big factor. So later on, I'll show you some images. In most cases, we ignore that heterogeneity. But for brain imaging, when the skull gets thick, then that becomes an issue. So this is the first functional photoacoustic image. We demonstrate this in a small animal. If you wiggle the whiskers on one side, the contralateral side of the brain will be activated hemodynamically. So the color shows you the blood concentration change, depending on which side you wiggle. And this is the first time that photoacoustics shows a high enough resolution image in a relatively deep region because light has to go through the skin and skull to reach the cortical region of the brain. And using standard confocal or 2-photon imaging, you cannot get this kind of images. So this work really galvanized the field since the publication of this work in '03, you can see the field has grown exponentially with a doubling constant of roughly 3 years. In '03, this was the smallest conference in Photonics West. Since '09, it has become the largest. So arguably, this is the fastest growing imaging modality, at least in optics. And it's growing even faster. Why is photoacoustics so exciting? Well, all modalities are complementary. So we're not replacing any modalities here. But photoacoustic tomography is the only one that provides you multi-scale imaging of organelles, cells, tissues, and even organs. And so this is the only multi-scale imaging modality that covers a wide enough range. Why is that so important? In the current practice, for organelles and cells, we use optical microscopy. So you're looking at optical contrast. For tissues and organs, we use non-optical imaging modalities, MRI, CT, ultrasound, whatnot. You cannot relate images acquired across length scales. There's a huge divide between the microscopic and the macroscopic worlds. Photoacoustic tomography may provide that unique link between these two big worlds. So my hope is that photoacoustic tomography can potentially enable systems biology research at multiple length scales, can accelerate translation of microscopic lab discoveries to macroscopic clinical practice. As you know, right now we have a lot of discoveries, but they don't translate into the clinic. Right? If we can double that translation rate or triple that translation rate, the impact will be enormous. I'm plotting the penetration in scattering tissue versus resolution. Each bar represents a real system implemented in our lab. So all of these are not hypothetical now. They are real. Forget about the actions. I'll get into some of those. All right. The first paper that was published in '03 demonstrating functional brain imaging was done through a Poorman's system where we had a single element transducer rotating around the animal, repeating the laser pulses. The data acquisition took more than 20 minutes. So now in collaboration with UConn, led by Dr. Zhu, we have a 512 element ultrasound array with 64 channels of A to D conversion. So with 8 laser shots, we can get a 2-dimensional image. That data acquisition can be accomplished within about a second. So we have a 1 Hertz 2D frame rate using this system. It's a lot faster than 20 minutes. This is the image acquired non-invasively using this system, imaging the brain cortex of a small animal. This is what you see in case you haven't seen a rat yet. Right? You shave the fur. So this is the skin. You can't see through the skin and skull and see the cortical structures using the naked eye because of light scattering. This is what we see non-invasively at this moment, right? And you can see all the cortical blood vessel structures with high definition. Of course, after we finished data acquisition non-invasively, we did something, open up the scalp, and you can see the cortical blood vessel structures quite well, and you can see how well they agree with each other, right? Now, the CM system, imaging system, is used to get a full body image. We're scanning along the trunk. Each frame in the movie shows you one cross-section along the trunk. And you can see some of the structures that are labeled here, like the kidney, the GI tract, the colon, right, and the backbone muscle as well. And you can see some of the boundaries which are quite sharp. And this image, this movie, was acquired only about a couple weeks ago. So we're still making progress here. This is very exciting, especially for biological research, for drug discovery, as you can imagine. We're using intrinsic contrast only. We can delineate the internal organs quite well at this type of resolution. We're always interested in bigger animals, eventually humans. Right? This comes back to your earlier question about the heterogeneity in the speed of sound. The skull has a speed of sound roughly twice as high as the speed of sound in soft tissue. Right. So that creates a problem. The good news is we have sufficient SNR. So this is an image of a rhesus monkey head with the skull intact. After we acquire the data, we open up the skull. You can see this is an open skull photograph, and compare them and look at some of the structural information. So this is very encouraging, but there's distortion due to the skull. And we have active R01 pursuing de-aberration, trying to correct for the aberration and see if we can get better images. But this is an important step forward. Now, photoacoustic CT also comes in linear geometry. There's this very nice scalability in ultrasound which applies to photoacoustic tomography as well. And this is why we can image at both the microscopic and macroscopic length scales. In photoacoustic tomography, if you look at the transverse or actual resolution, they're both inversely proportional to the center frequency of your photoacoustic wave. The penetration limit is also inversely proportional to the frequency. But if you divide these two, you find the ratio is roughly a constant. And very likely, that constant is about 200. That means you can count on roughly 200 pixels in the depth direction. Right? I'm very thankful that this constant is 200 instead of 2. We don't control any of those. These are determined by nature, by tissue properties. Right? If you get 2, you'll end up with 2 pixels in the depth direction. You won't have a good image. In collaboration with Philips Research, we now have adapted a clinical ultrasound machine for concurrent photoacoustic tomography. You can see this linear ultrasound probe linked with 2 optical for light delivery. Single laser shot will generate a two-dimensional image in the B-scan direction. Right? One of the dimensions is depth. Because 128 channels of ultrasound transducers will detect in parallel. So this is an extremely powerful system because our idea is physicians have access to ultrasound already. They feel comfortable with ultrasound. Now we tell them with a single switch, you can retain all the original ultrasound imaging capabilities, but we add you photoacoustic imaging. So you see different contrast mechanisms. You see optical contrast on top of the original ultrasound contrast. We're targeting a number of applications using this device. One of the applications is reporter gene imaging. You've heard of the fluorescence-based reporter gene imaging, and that allows you to image maybe a millimeter or 2 in the skin if you want to have a high resolution. Here we can image multiple centimeters and still have a sufficiently high resolution. Right? This can potentially extend the capability of reporter gene imaging. We're also targeting a clinical problem, breast cancer staging. The idea is to inject methylene blue dye, and the lymphatics will gobble up the dye molecules and accumulate at the central lymph node, the first draining node. We can see the dye accumulation very well because the dye is a great optical contrast agent. Then we guide a needle to take some tissue out of the central lymph node, do a histology on that, and see if there's any cancer at the central lymph node. You can also use ultrasound to guide the needle, but the contrast is much lower. It's about 1.2 versus the photoacoustic contrast of the needle, which is about 8.5. The standard practice right now for breast cancer staging is to inject radionuclides and optical dye. Then you roughly locate a position due to gamma photon emission outside the body. Then you cut open the breast. So it's a surgical procedure. Then you visually follow the dye until you see the node, dissect the node for histology. 80% of the patients who go through the procedure turn out to be benign. And this surgical procedure not only is costly, but has also caused some complications. Right? So if we can avoid that, turn that surgical procedure into a needle biopsy procedure, we can save a lot of problems. one of the key questions is how deep can we penetrate? And whenever you use light, that's the first question you ask. If you use the right wavelength, you can penetrate multiple centimeters as we demonstrate. This is still ex vivo. So we're using chicken breast tissue to mimic the physical property, the optical properties of human breast tissue. This experiment shows that we can penetrate about 7 centimeters. Right? But you can. I'm sorry?
What wavelength is that using?
This is a 650 red wavelength. There's this microscopic domain through photoacoustic tomography. Let me start with acoustic resolution version. In the CT version of using photoacoustics, you're counting on math to reconstruct the image. Each transducer is unfocused. Here, we're using physics to form an image. The transducer will be focused. Assuming there's an optically absorbing target, with fire laser pulse into tissue, you're going to generate a photoacoustic wave, and that's received by this focused ultrasound transducer. You get a time trace like this, so this target is going to show up as a spike due to stronger absorption. If you have a second target somewhere here, you would see a second spike. So this 1D signal can be converted into a 1D depth resolved image. Right? Because we can convert time into depth. Just simply multiply the time by the speed of sound in the soft tissue. If you transversely scan, you get a 2D image. That's a B-scan image. The 1D image is called A-scan. If you raster scan on the surface, you get a 3D image. This is the first 3D photoacoustic microscope, which was built in our lab. You start from a tunable laser system, route the beam through optical fiber. You see the beam coming out of the fiber, starting with a solid beam. We convert a solid beam into a hollow cone beam through this conical lens or hexagon lens. Then this component is shown in this close-up, which is really the key component here. The hollow cone beam is refocused down into the tissue. On the tissue surface, you have this donut beam. The core is dark because we want to minimize the surface interference. Then this focused ultrasound transducer is detecting the photoacoustic signal. You get a 1D image in the depth version. This head is raster scanned in this water tray, and we couple light and sound through this membrane window, and the tissue you want to image is placed underneath the membrane window. This was one of the first images acquired to demonstrate the capability of intrinsic contrast and resolution. So this is a photograph of melanoma. You can see a little bump here, but you don't see the depth. You don't see the surrounding blood vessel structure. Using melanin and hemoglobin as intrinsic contrast, we can see both. Here's a melanoma. You see the parallel blood vessel structures as you would in a physiology book, textbook. Except here, everything's done non-invasively. The intrinsic contrast is extremely high, as shown here, where in X-ray imaging of soft tissue, you're typically dealing with 1% soft tissue contrast. So that's why you often have to inject iodine-based contrast to see the blood vessels. Here we don't. The red blood cells are your endogenous intrinsic contrast. Human imaging is possible. So this is the area of the palm we imaged. You can see the blood vessel network extremely well. Again, without injecting any contrast agents. This is a B-scan image, depth resolved, showing you some of the standard structures in the skin. Nanoparticles are natural contrast agents for us, as long as they have optic absorption. So we work with our collaborator in our own department, Dr. Shah, who invented nano cages. They provide very strong light absorption as a good contrast agent. One step further, they targeted those nano cages onto melanocyte stimulating hormone receptors. By targeting the receptors, we enhance the contrast by a factor of 3 in comparison to the non-targeted nanocages. And we're also interested in monitoring therapy. Nanocages turn out to be a very good carrier for drug delivery, and we can monitor the whole process. We also miniaturized the probe for endoscopy. The key idea is this rotating mirror that reflects both light and sound. We use optical fiber to deliver light. The light is reflected by this mirror surface, delivered onto the tissue, which will generate photoacoustic wave. The acoustic wave will come back and reflected by the same mirror surface, detected by this ultrasound transducer. Now, sound transmission does not like air. So this chamber is liquid filled, but this micromotor does not like liquid. So this side is hermetic, air filled. The two chambers are separated, but the torque is transferred through this pair of magnets. So this movie is showing you the device in action. You can add a mechanism to pull back the device as well. So this is a set of in vivo images acquired from a rabbit esophagus. As you pull back the device, you get a series of circumferential B scans. This is a photoacoustic image. Concurrently, we get an ultrasound image. You can see some of the structures quite well. And this is a 3D rendering of the same data set for both photoacoustic and ultrasonic images. An important feature here is that we can penetrate 7 millimeters in the current setup into the GI tract. If you use standard optical endoscopy, your penetration is limited to about 1 millimeter. In many cases, there are deeper lesions. The GI specialists want to penetrate about 3 millimeters or so to capture all the other deeply seated lesions. And this device is the right target device. Photoacoustic microscopy also works within the diffusion zone, within about 1 millimeter in penetration. And we call that optical resolution photoacoustic microscopy because within the first millimeter of depth, you can still optically focus. That gives you the transverse resolution. The key component in this device is to combine the optic axis and acoustic axis. We use this light-sound combiner made of 2 prisms separated with a gap. The gap is filled with liquid. So along the optical axis, the refractive index is well matched, which gives you very high transmission of light. However, at this solid liquid interface, the acoustic impedance is highly mismatched, which gives you a very strong reflection of sound. That's why the photoacoustic wave coming back will be reflected toward this ultrasound transducer. A single laser shot will give you a 1D image depth resolved. Then you can raster scan his head to get a 3D image. This is one of the first generations which gave you about 5 micron transverse resolution. As we know, the smallest blood vessels, namely capillaries, are roughly 7 microns in diameter. So 5 microns, you can detect single capillaries. And we imaged the same animal over a period of 60 days just to monitor the angiogenesis process. We work with Dr. Arbeit. He used to sacrifice animals at different time points to get the same data set. He had to deal with intersubject variability. We don't have that issue because we're monitoring the same animal. You can see increasing vessel density as well as vessel tortuosity. Yeah, please.
How do you induce angiogenesis?
And this is a transgenic model that has angiogenesis. And this is a rendering in 3D showing you the data set. We increased the resolution down to 2.6 micron, roughly about a factor of 2 improvement. And so that allows you to resolve even single red blood cells. And this is a 3D image showing in 2D projection view of the entire mouse ear without injecting any contrast agent. To do the same thing with micro CT using X-ray, you have to inject iodine-based contrast. Even with the contrast agent, you still miss capillaries. So this is a very natural tool for blood vessel imaging down to the smallest capillaries. You can zoom into a small region to see the details. You can see a capillary bed, a bunch of single capillaries. All these single dots are single red blood cells. We improved the resolution down to 0.9 micron. At that resolution, you can even see the biconcave structure of a single red blood cell, as shown right here. This was done in vivo. Another interesting phenomenon is called intussusceptive angiogenesis. Right, this is the first in vivo image of intussusception. All previous studies were based on electron microscopy. That means you have to sacrifice the animal in order to get an electron microscopy image. Right. What's going on here is you see this blood vessel with a hole in the middle. Unlike sprouting angiogenesis where you grow a new vessel altogether, here you split an existing vessel into two, and we're monitoring that process in action. So hopefully this is going to reveal some new biology. We work with a neurologist who is interested in studying Alzheimer's disease, plaque formation, and he can tolerate the removal of the scalp in some animal, but not the skull. The minute you remove the skull, you can cause artifacts. By going through the full thickness of skull, we can form a very detailed image using this device. If you were to use confocal or two-photon microscopy, you got to thin the skull or remove the skull to get the same type of images. So you can see how powerful this is in biological research. And we can stain the structures in the skull by using methylene blue. And of course, we can tune our laser wavelength to target blood versus methylene blue. Human imaging. So we image our own finger cuticles. You can see all the capillary loops. Again, these are the tiniest blood vessels. This was done in vivo, of course. In addition, because oxy and deoxyhemoglobin have different colors, you know that in everybody's blood, you got two forms of hemoglobin. Right? They have different colors. By tuning the laser wavelength, we can separate the contributions from both forms of hemoglobin and quantify the concentrations of both, from which we can quantify the oxygen saturation of hemoglobin that runs from 0 to 100%. And the color here will show you the distribution of oxygen saturation. Let's zoom into one of the capillaries. You can see here on one side it's all red, on the other side is all green. That means one side has high SO2, the other side has low SO2. What does that mean? Most of the oxygen is dumped at the tip. This is a new physiological observation. We really try to push the resolution within the diffusion or diffraction limit in this case. You can use a very high NA. In fact, in this case, we use the water immersion NA. We push the resolution to 220 nanometers. And this is a point spread function taken out of a single nano cage, single nanoparticle. Right? So the half width and half maximum is about 1 or 10 nanometers. We can target DNA and RNA absorption to see the cell nuclei directly, because we know cell nuclei has an increased concentration of DNAs and RNAs without any labeling, right? So we're essentially doing label-free in vivo histology. As you know, in standard histology, what does it mean? The physician will take out some tissue out of you, then section it, then stain the section, then look at it under a microscope. The whole procedure is invasive. Right. Now we can do the same thing in vivo by just targeting the DNA and RNA absorption. Right. You see this same piece of tissue without labeling, and we get an image. Afterwards, we use standard HE staining to get a histology image. There's a one-to-one correspondence between the cell nuclei distribution.
And the DNA and RNA absorption is narrow enough and specific enough you can target it precisely?
It's narrow. It's definitely narrow enough. Yeah. So if you have other absorbers competing, you can do spectroscopy. So you tune your laser wavelength, then you can separate the contributions. But it's obviously not a problem here. So. Not only can we image the total concentration of hemoglobin, the oxygen saturation of hemoglobin, but also we can image the blood flow using the photoacoustic Doppler effect, which was discovered by our lab only 3 years ago. And we can also image the cross sections of blood vessels. We can definitely image the volume of interest. Now, photoacoustic tomography is the only modality that allows you to image all 5 parameters intrinsically, which will allow you to quantify an important parameter called metabolic rate of oxygen. Right? This is an extremely important parameter because, as we know, cancer has a problem with metabolism. Metabolism has gone awry. If you can image the metabolic rate of oxygen, you can potentially image cancer early, intrinsically, without injecting any toxic contrast agents. So we're very excited about this. In fact, later on, we found out something very surprising. You talk to biologists, most of the time you'll hear that cancers are hypoxic, meaning within the cancer there's a lack of oxygen. But that's not true in early stage cancer. You can see here this is a cancer that has hyperoxia. It has excess of oxygen in the bloodstream. Even though the cancer has a very high metabolic rate of oxygen in the beginning. The cancer is consuming more oxygen, but how come it has excess of oxygen in the blood? This is very counterintuitive. The reason is that you have a much increased supply of blood due to angiogenesis. You've grown a new set of blood vessels that supplies so much oxygen. Even though you're consuming more, we still have more oxygen. So from this data, you can argue that MRO2 is a much more fundamental parameter than just oxygen saturation, because oxygen saturation alone doesn't tell you the metabolism. Okay. A quick discussion. Right? Besides light, you can also use microwaves or radio frequency waves to excite acoustic waves. Right? So that allows you to get different contrast mechanisms in the microwave regime, primarily water. Right? So we're interested in pushing that direction as well. Here you see the laser is gone. We're using a microwave generator to excite acoustic waves. And we moved the device, which is small enough, to MD Anderson Cancer Center, imaged a few mastectomy specimens. You can see the tumor quite well. This is like a 5:1 contrast. This is like a 3:1 contrast. Right? So we're excited in pushing that direction further. I'll use one slide to introduce another technique we developed recently in our lab called time-reversed ultrasound encoded optic focusing, or true optic focusing. You know, we know that it's really hard to focus light beyond 1 millimeter in depth. In order to do that, we say, well, what happens if we generate a guide star? In astronomy, folks have been using that technology for a long time. They use a naturally occurring guide star or artificially generated one that allows you to quantify the wavefront. You measure a distorted wavefront, you say, well, it should be a spherical wavefront, but now it's distorted. You know the gold standard, you can correct for it. That allows you to get a good image. But in biological tissue, we don't have a guide star to begin with. You can implant one, but that's invasive. And you implant to a given position X, Y, Z, it's fixed. You can't move it unless you plant another one. So that's a problem. What happens here is, let's say, well, let's use ultrasound. Because ultrasound scattering, again, is so low, you can point your ultrasound focus to anywhere you want. The ultrasound is going to modulate your light by shifting the frequency. Somehow the movie is not running. So this is the forward process. You send your light in. When the light goes through the ultrasound focus, it'll shift the frequency, so it'll change color by a tiny bit. And outside we have a phase conjugation mirror that's going to return the light backwards, time reverse the light. And those frequency shifted light will be the light to be captured. So you only return the frequency shifted light. And that part of the light will be going back to the ultrasound focus. So now you have a virtual guide star. You're able to focus light into a scatter medium. Now, if you don't use this concept, you can focus light through a piece of tissue. But this is much more useful if you can focus light into a piece of tissue. And this is a very new concept. It's still early. We're pushing this further. Hopefully, one day we'll find applications. Now, if you can focus light into biological tissue, you can impact everything in biomedical optics, from imaging, sensing, manipulation, or therapy. One example of manipulation is optogenetics. Example of therapy would be photodynamic therapy. If you can focus light deeper, you can deal with deeply situated areas. Right now, optogenetics only works on the surface, or you have to use a fiber to deliver light invasively. In summary, photoacoustic tomography can be achieved by optical excitation and ultrasonic detection. The diffusion limit has been broken. Super depths up to 7 centimeters have been reached. Single capillaries or single cells can be resolved in vivo. Up to 220 nanometer resolution has been achieved in the transverse direction. Multi-scale imaging can be achieved by scaling the depth and resolution, by varying the bandwidth and center frequency of the photoacoustic detection. Photoacoustic detection is background free, meaning when there's no optic absorption, you get no signal. From engineering perspective, we know that's the most desired situation. In fact, photoacoustic tomography has the highest sensitivity to optic absorption among all modalities. It's at 100%. Either non-fluorescent or fluorescent pigments can be imaged. If you depend on fluorescence imaging, you got to have a reasonable fluorescence quantum yield. Not all molecules can provide you that, but essentially all molecules are absorbing. It's a matter of finding the right wavelength. Multiple chromophores can be resolved spectrally. And functional imaging is achieved using endogenous contrast like hemoglobin or melanin. And molecular imaging is enabled by targeted contrast agents like nanocages. You can also use organic molecules. Reporter genes can be imaged as well. Now, using the Doppler effect, we can image blood flow. That's another functional parameter. The imaging rate can be fundamentally very high. In practice, we demonstrate 50 hertz online, 249 hertz offline, and we don't see speck artifacts. If you work on OCT, you'll realize that in a scatter medium, you'll see speck artifacts. In ultrasound imaging, you face the same issue. We only use non-laser radiation, so it's very safe. The systems are relatively low cost. We do require contact detection through either liquids or gels. Cavities and very thick bones can present challenges to ultrasound transmission, but we're finding ways to circumvent that problem as well. We do have this book which contains a chapter on photoacoustic tomography if you're interested in finding more details. And there's also this edited book. A lot of the leading experts in our field have contributed to this book. Finally, let me give credit to my lab members. All these colored names are major contributors to this talk. Thank you very much. Yeah, please.
In submicron imaging, when you're focusing with an optical beam into a small spot, to do an A scan, you have to scan the spot in the Z dimension.
If you want to have a long depth range, you do need that.
Well, if you want any depth range, don't you have? I mean, because it almost
Yeah.
very quickly.
The focal zone is very short. You're right. Yeah.
So in like the images you were showing, is there a Z scanning going on?
We didn't do a Z scan in that case because the feature doesn't have a huge depth range. But in reality, if you want to image a three-dimensional structure, you may want to do a Z scan in addition.
So So when you showed those 3D images, it looked like they have lots of depth to them. Was I not looking at those correctly?
For the extremely high NA, the depth range is relatively small, but
How small?
so that's probably tens of microns.
So you can go much more than the focal depth of that.
Well, your resolution will not be as good as exactly at a focal point, right? So it's going to degrade, but it's not going to go away totally.
So if
Yeah.
you did need the Z-scan, you would only Z-scan the optics, not the acoustic, if there was an acoustic focusing.
It'll be less sensitive to the acoustics, because our acoustic focal zone is very long. It's more or less like 15 microns. So yeah, you do the optical Z-scan. Yeah, please.
What drives the choice of wavelength?
So there are different chromophores in the tissue. You have hemoglobin, melanin, maybe lipids you want to image, or water. In most cases, you want to find the absorption peak of the chromophores. For hemoglobin, you got multiple peaks. Most of the time, we use the Q band instead of the 3 band. The 3 band is 10 times more absorbing than the Q band, but it's too absorbing. That means your penetration will be very limited. So there's a trade-off where you can find a happy medium. You can spectrally unmix the contributions of different chromophores. Like the oxy and deoxy hemoglobin molecules, they overlap substantially over the entire spectral range for absorption. But if you use multiple wavelengths, you get a set of equations, then you can unmix the contributions. Yeah. We haven't done bezel beams. We talked about it, and then there are people who are pro and there are people who are against in our lab, and in the end we decided not to pursue because of the side lobe issues. Yeah, but it's certainly an interesting concept. Yeah, please.
So in your these conjugation scheme, if I understand correctly, the size of the spot of the focal spot is limited by the.
Correct.
So what kind of applications can you imagine?
Initially, we're targeting relatively deep imaging. So breast, for example, you can still focus using like even 10 megahertz. And so lambda would be 150 microns. And for breast imaging, if you can get a millimeter resolution, it's very good. Now, if you can get hundreds of microns, it would be even better. Now, acoustics is highly scalable. If you don't want to penetrate so deep, for breast imaging, you're talking about centimeters. If you're within a centimeter, you can probably increase the frequency to tens of megahertz, and that'll give you a smaller spot size as well. So it's a scalable problem. Yeah.
Any other questions?
Yeah.
Actually, yeah. So some of the systems that you showed, you were putting in light and you collected back,
Yeah.
say, a time signal.
Yeah.
To find a delay. And then you said you scan you would scan transversely, and I never really saw what the mechanism you were using to scan transversely. I'm just curious.
You can mechanically scan. So we just mechanically scan the whole scan head.
Okay.
So at every new position, you fire laser pulse again, you get an E-scan, 1D image. Yeah, you make a composite image at the end. And you got a 2D array of 1D images. So you can also electronically do it. Right? That's basically what we do in photoacoustic CT. And we use an array of unfocused transducers, detect all the acoustic signals, then synthetically focus to every spot within the plane. So I understand at least one of you is doing the optical counterpart of that. So, yeah, please.
So, in photography, you use the array of detectors.
Yes.
Do they have a frequency?
Yes.
So, how do you decide the frequency?
So again, that's a scalable issue. With ultrasound, if you want to have very high resolution, you want to use wideband and high center frequency. But At that high frequency, you have stronger ultrasound attenuation, so your depth of penetration will be limited. From a practical perspective, the first thing you want to do is to find the depth you want to penetrate. Then that defines the frequency you can use, the highest frequency you can use. From the highest frequency, you can figure out the resolution you can afford. So everything starts from the desired depth. That's why we have, different systems. They all target different depths. Starting from like a millimeter penetration, up to like multiple centimeters. Does it make sense?
So, one more time for questions, a little request. For those of you that are not registered for the COSI seminars as a class, we would like to know who is coming for us attending. So we have a list here. If you want to put your name in there, we will know who is coming, and we will tailor the seminars also for other attending people. In each bench there's a list or here in the front. So thanks everyone.
Thank you all.